Hello. Let me begin by expressing my gratitude to the gracious host and all the distinguished guests gathered here to discuss about the advanced imaging in the various physical and biological application. My name is YongKeun professor in physics, and today I'd like to talk about quantitative phase imaging, three-dimensional quantitative imaging, and which is called the holotomography, and how we can use HCT and the artificial intelligence for three-dimensional biology and various new field of bioimaging. To make a long story short, I'd like to first talk about these images, which you can see here in the title slide. It is three-dimensional label-free images of embryo. The size of embryo is about 150 micrometer in diameter, and without any staining, no fixation, any preparation protocol is needed to see the high resolution, three-dimensional, label-free, non-destructive images like that. So I'd like to talk about imaging paradigm in bioimaging. As we know, the bright field microscopy has been used for the microscopic object. But due to the little scattering, so many biological components, it's not very clear using the bright field microscopy system. That's why Janicke came up with a very clever idea of using phase contrast with interference phenomenon to increase the imaging contrast. So by simply inserting the plastic filter into bright field microscope, you can significantly increase the optical contrast for unlabeled sample images. And the modern biology is driven by the molecular specificity. So by using the exogenous labeling agents such as the molecular proof or molecular the staining agents such as fluorescent proteins, you can see individual proteins and fluorescent labels object inside of cell, which has opened doors to molecular biology and molecular diagnosis in application. And now we see the new eras where we have to see three-dimensional architectures of cell and tissues, such as organoids, which is three-dimensional in vitro models of organs using live cell. And we also see the many interesting applications using the stacked layers of the cell complex, such as iPSC colonies and 3D histopathologies and so on. So we need to have three-dimensional tomographic imaging capability. And then in order to use those samples for therapeutic and the reproductive process, we have to make them alive and intact without using any exogenous labeling agent. And that's why we are working on label-free imaging technique. In particular, we are interested in using the reflective index as imaging contrast. So by using conventional methods such as personally labeled cell, by using confocal microscopy systems and so on, you have to prepare a cell a few days before your measurement by transfections and the washing out and sometimes the fixation and optical clearing index matchings are required. Even though you may get beautifully prepared the fluorescently labeled cell, you have to discard cell after your measurement because cell has been genetically modified and sometimes fixed and killed. And obviously, you cannot apply this labeled cell for therapeutic application. You cannot put those labeled cells into human body. Situation becomes more complicated if you want to measure three-dimensional this fixed samples, such as the organoids or the three-dimensional the histopathological samples. Conventionally, what they do after fixation and followed by the serial sectioning, individual sections are labeled by using staining dyes such as H&E, and each 2D thin slice are measured by using whole slice scanners. And then they do synthesize in order to get three-dimensional volumetric representation. But due to this in the sample preparation and serial sectioning, sample has been sometimes distorted, and you cannot recover after those physical damages. In addition to the very long preparation time and the cost. And using label-free quantitative phase imaging technique or holotomography, it only takes few seconds in order to measure whole volume of imagings, like covering few hundred micron by few hundred micron by few hundred micrometer. And since we are using intrinsic imaging contrast by using refractive index, no preparation is required. You can just simply put your samples into instrumentation, and then you can get morphological information of cell and tissues in 3D. And then after measurement, you can just put the samples into therapeutic application. And now we can measure organoids and 6 samples of 250 micrometer in diameter with a resolution go down to 150 nanometer. And HET is principle, it's like the optical analogs to X-ray CT. As we know, X-ray CT use the inverse wave propagation, inverse scattering problems. So by illuminating the multiple illuminations through human bodies, you can measure multiple 2D diffraction intensity images from which you can reconstruct three-dimensional distributions of the human body in terms of X-ray observability. We can do the same thing by using light and by illuminating the beam passing through cells and organoids, and we measure holograms in terms of consisting of both amplitude and phase information. And then by illuminating with various illumination angles or controlling the wavefront information, you can measure multiple 2D holograms as a function of illumination angles. And then by inversely solving wave equation, you can reconstruct three-dimensional tomograms in terms of refractive index. The first theoretical paper was written by Professor Emil Wolf in 1969, and after that, many experimental and engineering optimization has been done for the last few decades. And in order to give you the big pictures, there are the bodies of the label-free imaging techniques such as phase contrast, DIC, autofluorescence, LAMA. They're all great techniques. And among them, if you utilize refractive index as intrinsic imaging contrast, this field is called the quantitative phase imaging, or QPI. And as a subset, if you could reconstruct three-dimensional distribution of refractive index, that is called the HET. And there are many different techniques in terms of realization, like optical diffraction tomographies or refractive index tomographies, Fourier type array tomography, and so on. And this is like the term to just include this field. Okay, so I don't have time to go through all the different variations of technique to realize HET, but in terms of the illumination source, you can think about the use of coherent laser beam, or you can use the temporary low current light, and you can also think about especially low current light and so on. And depending on the kind of technique was applied to retrieve both amplitude and phase information, there are like the magenta type interferometric microscopy system, or you can use the reference-free system to measure diffraction patterns from which you can retrieve both amplitude and phase using various other techniques. So to do that, you have to have, regardless of these configurations of optical systems or the light source, the essence is that you have to retrieve both amplitude and phase information, and then you have to illuminate with various illumination the mode so that you can inversely solve wave propagation equation to reconstruct three-dimensional distributions of the optical scattering potential or refractive index distribution. The physics is same, but realization depending on how you can define the different configuration. And these are the representative technique. You can use laser beam and magenta interferometric system where you can measure specially modulated holograms as a function of illumination angles, and then you can retrieve three-dimensional distribution of refractive index by applying the diffraction theorem to get the three-dimensional distribution of scattering potential. Or sometimes you can use the incoherent light source such as LED, and by controlling illumination patterns, and then you can measure multiple transmitted diffraction patterns in terms of the intensity distribution, and then you can apply the algorithm to get three-dimensional distribution of refractive index. For further information, you may want to refer to these review articles. And one of the contributions that our lab has they've applied is that what we have they came up with an idea is we were able to retrieve both the amplitude and phase measurement without having the reference arm and with like very simple configuration. And we can, when you used the spatial domain Kramer-Kronig relationship, we were able to retrieve both amplitude and phase distributions from single measurements of intensity patterns. And then by controlling the illumination angles, you can also realize holotomography system. And then we were able to reconstruct three-dimensional distribution without having the complex and the sensitive reference dependent on the setup. So using HET, you have several benefits. So first, no labeling because we use reflective index as imaging contrast. So no phototoxicity or photo-bleaching issues because we do not use any exogenous labeling agent. And there's no limited type of measurement. And our speed of measurement is about 100 times faster than confocal fluorescent microscopy system in order to cover the same field of same volume of interest. And our resolution go down to 150 nanometer, and we can measure the sample thickness of 250 micrometer. And I'll talk about quantitativeness in the following slides. So this field, first, they studied the biophysical and simple configuration, simple structure cells such as red blood cell, and now is moving from like the biophysics to cell biology to developmental biologists and organoids and tissue sensors. It's expanding by having the wider ranges of spatial and temporal scales of measurements. So what you are seeing here is like the three-dimensional tissue culture, but this is the neuron cell. You're watching not artificial neural networks. This is real biological neural networks. So you can see the individual neuron cells are connected by the axons and dendrite structures. And you can see this neuron is trying to make a new connection. This is remodeling or plasticity happening in your brain right now. So you can see how actively, how gently we can measure this highly sensitive cell in 3D and time lapse with high resolution. Since our resolution go down to 150 nanometer, we are able to observe very sensitive and subtle subcellular dynamics. You're watching the the fusion and fission dynamics of individual mitochondria in live cancer cell line. And you can also observe there's a nucleus membrane and nuclear lipid drumlet and other membrane fluctuations are measured without having any preparation and very high spatial and temporal resolution. Since we are relying on the use of the intrinsic optical parameter, refractive index, you can measure the cell dynamics for forever. So you're watching the same field of view and same sample for 42 days. Because if you combine the system with the incubator, you can just keep the cells happy and alive by controlling the temperature, CO2 levels, and so on. And then you can see the how individual cell, which is called the fibroblasts, are differentiating into the different types of cell, which is called adipocytes. And adipocytes are known to generate the lipid drumlet inside. So you can see the formations of individual lipid drumlet having high refractive index over time. And then at the days of 42, we were also able to measure the large field of view. So this is like 4 millimeter by 4 millimeter. It's again 3D. But since our monitor is 2D, we only visualize the middle section. But nonetheless, you can see the individual, the lipid drumlet structures inside of cell. And if you have the small lipid drumlet, that is called the brown fat. And if you have the white, if you have the white and the big, the lipid drumlet is called the white fat. And you can also see other subcellular organelles such as mitochondria and nucleus membrane intact in 3D. Like Google Map, you can just go all the way down and all the way back. And now we can also measure 3D biology, such as the organoids. So this is liver organoids, and size of organoids is about 150 micrometer in diameter. It's like a shell shape. For visualization purpose, I only visualize in the bottom half. But you can see individual cells and their subcellular organelles. But important thing is they are live. So you can see how cells are dividing, and sometimes they are being they've been killed, and you can see there the timeless behavior over like hours and days and sometimes in the weeks. And now we are able to measure 36 samples. So this is the human intestinal organoids, again, label-free, live. And we can measure up to the thickness of the 500 micrometer right now by considering the multiple scatter light. But we are trying to improve the penetration depth further and further by pushing the limit of the optical imaging capability so that we can see this technique being used in the biotech and pharmaceutical companies and hospitals. So we have commercialized this system by the company Tomocap that we have co-founded, and by combining spinning disk confocal modules to HET, we can perform correlative imaging. It's like PET-CT. By simply putting the CT, if you combine PET to CT, then you can retrieve both structural information from CT and molecular specific information from the PET. We can do the same thing by combining HET and 3D fluorescence. So you are watching here 3-dimensional shape of the organoids observed by using HET together with the 3 multi-channel the 3D fluorescence, so that we can do correlative imaging analysis. Although this field is very new and this technique has been introduced to the field of the bioimaging very recently, but there are many active users in many leading institutes, and they have published many exciting papers by using this holotomography system. For the last 1 year, we saw the 3 science papers and 1 Nature Cancer paper. So we are now the new areas by the translations of the lab developed the holotomography system, and is widely and actively used by cell biologists and clinicians. And recently we realized in order to increase the applicability of HET, we need to use the AI. The first, we are dealing with the large data size, high resolution, 3D time-lapse. We can easily generate the terabytes of the data size. And then we are generating the new information that people haven't seen before. So I think it is crucial to combine HET and AI. So by doing that, we can do the several the opportunity. We can do the research of areas can be opened. For example, we can increase the imaging capability and imaging quality. We can do that. And then we can do segmentations of cell membrane and subcellular organelles as segmentation. And we can do classifications of cell type. And then based on the morphological shape observed by HET. And I'll talk about the virtual staining in the following slides. So this is examples of how AI can be used to perform the high throughput segmentations of cell membrane. And this is the interaction between CAR T cell and attacking the tumor cancerous B cell. And then by applying AI, we are able to segment out and perform pseudo coloring to make sure that which one is the effector cell and which one is target cell. Again, the samples are label-free and happy, but we have used the artificial neural networks to segment out which one is the killing the cancer cells, which one is the immune cell, and which one is the cancer cell. And we have further improved the capability, and now we can do the automatic segmentations of major subcellular organelles such as the nucleus and nucleoli and membrane and the lipid pyramid and so on. Those can be done by simply applying AI model to this label-free, the reflective index distribution data. The second application is segmenting the classification. It's like finding which one is cat and which one is dog. So after measuring these bacterial movies, we realized that we may be able to differentiate the different the bacterial type, because we can access the internal distributions of the proteins in the biomolecular content by retrieving 3-dimensional distribution of refractive index. So in collaboration with the Samsung Medical Center in South Korea, we were able to measure 19 different bacterial species. And each species, we have measured about 300 to 500 bacterial cell using HET. It doesn't take too much time because our measurement speed is very fast. And then after training the convolutional neural networks, and once the network is the network training is done, we apply these networks to unknown samples. For unknown samples, we measure single bacterium and test the networks to see which one is the species of bacteria. And accuracy was 82.5%. But if you apply ensemble average after measuring, like, let's say 5 bacteria from one unknown sample, accuracy goes very close to 100%. And if we do the optimizations of the algorithm, accuracy goes above like 95 and 97%. Virtual staining. So every technique has pros and cons, right? And the benefits of HET is label-free, high resolution, and the three-dimensional imaging capability, which is good. But one of our weakness is that we do not provide molecular specificity. What I mean by that, even though we can see the major subcellular organelles without any preparations of the sample, but if you ask, where is actin cytoskeleton? We have no idea because refractive index does not have molecular specificity intrinsically. And that's why many of the users and collaborators are still prepare cell by using fluorescently labeled technique, such as fluorescent proteins, so that they can measure both HET and multi-channel fluorescent images. So they do provide those information to journal articles publication. So we wanted to remove the need for the staining. So what we have done is we have used the artificial neural networks to virtually staining the fluorescent signals. What we have done is that for training purpose, we have prepared the fluorescently labeled cell. And for same cell, we have measured the holotomography data. And then we also measured multi-channel 3D fluorescence. And then we have trained the neural networks. And after training is performed, you do not have to label the cell anymore. So by measuring the unlabeled cell using HET and applying the networks, you can generate the artificial fluorescent data set. And accuracy was very high. So this is the for one example. Cancer cell line, this is real measurement, and the right panels are all virtual staining data. So you can generate the spatial biological information without doing the labeling or staining. So now we wanted to move further by expanding this application to the histological application. So there are like the big move in the cancer research from 2D to 3D. The idea is that tumor is 3D, but why should look at these tumor tissues by dissectioning, or why we have to look at the 2D? It has been used for more than like centuries, but if you just do the 2D sectionings and measuring tissues in slides, 2D slides, you miss important tumor microenvironment information. And that's why many research groups are employing the many different techniques such as micro CTs and the light microscopy systems and the serial sectioning. Those are all technically important application, but we wanted to use HET and virtual staining. So to make a long story short, we have prepared the 6 samples. It's like the 50 micrometer tissue samples from various cancers. And then by applying AI, and this is the virtual H&E data without serial sectioning, no staining. If you're trying to staining these 6 tissues, because of different diffusivity of the hematoxylin and eosin molecules, you just mess up the samples. And then there's no 3D absorption imaging technique in the available. So this is what you're gonna get. You do not get 3D data, and the coloring is totally different from what you can expect from the 2D scene slide. But using this HET and virtual staining, we can generate artificial staining, virtual staining or digital staining of H&E to the volumetric tissue samples. So far, I have talked about how what is the principle of HET and how we can use AI to improve the capability of HET, but those are all done in the optical domain. And if you look at the the first paper of the Emil. This equation and the principle is based on the inverse scattering of the wave propagation or Helmholtz equation. It should work for any wavelengths from UV, IR, and any electromagnetic wave. It should work. So as the starting point and pivoting in the study, we apply the technique developed in our lab in optical domain to X-ray. So for the last few years, we were able to demonstrate that we can improve the imaging resolution by using holographic technique and scattering the behavior. And we apply the Kramer-Kronig based the tomography approaches originally developed in the optics domain. We apply it to the X-ray domain. And then we are also able to measure the time lapse, the light field measurement, so that we were able to characterize the field informations of the FPL X-ray pulse information. And furthermore, what we have done is the reconstructions of the refractive index distribution by inversely solving wave equation. But as you know, the refractive index is not the full descriptor between to explain the light matter interaction. To fully describe, you have to have dielectric tensor distribution in 3D. Then you have full information how your matter, considering their orientations, molecular orientation, and interact with arbitrarily polarized light in space, in the propagation. You can fully describe. So we looked up the literatures and realized that there's no theoretical and experimental demonstrations of the measuring three-dimensional distribution of dielectric tensor tomography. So we have started from the theoretical development, and we are also able to demonstrate this technique can actually measure the three-dimensional distribution of dielectric tensor, which is dielectric tensor tomography system in the optical domain. So this is the liquid crystal, the networks. We were able to see the topological defects. And after that, we further improved these capabilities. So recently we have developed the algorithms and regularizations and further and further. And now we have further improved in quality. So we were able to measure three-dimensional the tissue sample structures, and we also measure very interesting biaxial and the uniaxial the crystal structures. So this is the ongoing studies, and we like to use this technique to characterize both biological and non-biological samples. So to summarize, I have introduced the principle of the HCT, holotomography, which is the optical analog to X-ray CT. And then it has several benefits. For example, label-free, three-dimensional high resolution and long-term measurements and quantitativeness. And then we also briefly introduced that by combining HCT and AI, we can do improved image quality, and we can do segmentation and classification and virtual staining. So with that, I'd like to thank for your attention, and I'll be happy to answer to your question. And one quick announcement is that you can use this QR codes to download the PowerPoint slides and for your information and education purpose. With that, I'd like to thank for your attention. Thank you.